Hello my dear students. Today we are going to discuss on our next topic, respiration. Another very important life process. In our first class we discussed regarding the photosynthesis, that means the formation of the food. Second one is the digestive system, means the breaking of the food. So first we got to know that how foods are prepared, in second how foods are broken. So what for? Because we need energy. For any of the lively activities we need energy, and only one process to get the energy is the respiration. Means we need to break this substrate. So when we are talking about respiration, it's one kind of metabolism, and we will say it is catabolic or anabolic, means making or the breaking. This is the breaking, means it's a catabolic process. In this catabolic process, what we are getting, first the C6H12O6, the glucose is going to be cleaved in first pyruvic acid. After that there is another set where pyruvic acid again broken within the Krebs cycle. So let's have a look today in this. So today our topic, respiration. To get the energy. Here we are having the respiratory substrate. Which one is the respiratory substrate? Of course the glucose. This is the reaction C6H12O6, which is going to be combined with the 6O2, and it will release ultimately this and energy. But it's a long process, and it is not going to be done only within the within a particular cell part. So respiration, now we can consider this respiration in two halves. One is the anaerobic respiration, another one is the aerobic respiration. In anaerobic part we are naming it the glycolysis. It is also known as the EMP pathway. Why EMP pathway? Emden, Meyerhoff and Parnes, three scientists, Emden, then Meyerhoff and Parnes, they discovered. Where it is performed? Place of occurrence, cytoplasm. What is the substrate we are having here? Substrate is glucose. And what is the product? This glucose is cleaved to formation of the glucose 6-phosphate, then fructose 6-phosphate, then fructose 1,6-bisphosphate, then 2-triose phosphate. Means this triose phosphate name is either glyceraldehyde 3-phosphate or dihydroxyacetone phosphate. Ultimately it will form that pyruvic acid. Which one is the ultimate product of the glycolysis which is happening in the cytoplasm? What we need to remember here? Glycolysis name, EMP pathway, cytoplasm, and this pyruvic acid. This part we don't need right now, but if you want to know, I can discuss in my next class. So whatever, the glucose triose phosphate, then pyruvic acid now formed, but it released very least amount of the energy. Only 2 ATPs are released. Where we are getting it? In eukaryotic and in prokaryotic. So in eukaryotic cell as well as in the prokaryotic cell, we are going to see this glycolysis is going to be performed. Here very meager amount of the energy we are getting, only even less than about 7% of the energy that within this glucose is going to be cleaved here. means less than 7% energy we are getting. If we want to get all the energy from glucose, we need to go to the next hub. Means that name is the Krebs cycle. Before that, anaerobic respiration types. Let's have a look. Anaerobic respiration again can be of two types. What? First one is in lack of oxygen or in absence of oxygen. Both the things are not same. Means here oxygen not at all needed. Means in yeast fermentation. Okay. And here in muscle cell. In muscle cell the product we are getting the lactic acid and very small amount of energy. Anaerobic respiration started from C6H12O6 as glucose is C6, and here from we are getting the lactic acid 2, 3 carbon. Yeast here from we are getting in absence of oxygen, no oxygen needed, here we are getting CO2 plus ethanol C2H5OH plus energy. Okay. Here pyruvic acid present. Now you can relate, this is a 3 carbon compound. I am repeating this part. Anaerobic respiration started from glucose 6 carbon. Meanwhile we got pyruvic acid 3 carbon. So how many pyruvic acid we will get? 2 molecules. Now each pyruvic acid, if in muscle cell lack of oxygen, it will release lactic acid. It is also 3 carbon. So no CO2. 3 carbon and 3 carbon, and energy very less amount. Here we are getting in absence of oxygen, here we are getting yeast fermentation, CO2, C2H5OH energy, 1 CO2 and here again C2H5 means 3 carbon balanced here from. Okay. Now let's move to the next hub, means the Krebs cycle. In Krebs cycle here, before that one step was there. What is the step? First this pyruvic acid is going to form the acetyl-CoA. Acetyl-CoA, it is not a part of Krebs cycle. Krebs cycle is after this. Okay, so pyruvic acid is going to be transformed into acetyl-CoA with help of pyruvate dehydrogenase, some ions, magnesium, NADH will be there, whatever, acetyl-CoA. This acetyl-CoA will now react with the oxaloacetic acid, a 4 carbon component. This is 2 carbon. So result is the citric acid. How many carbons? 6 carbon. With some intermediate product, it will form the alpha ketoglutaric acid. It is 5 carbon. So here also CO2 released. Then it will form ultimately succinyl-CoA, succinate, this. Then it will form, this is 4 carbon. Then it will again form the fumarate, means fumaric acid. Then it will form the malate or malic acid. This is the Krebs cycle. It will release all the energy. How? Here from we will get so many numbers of the NADH, FADH or FAD, and with this we are getting the one GTP or ATP. Ultimately they will enter the ETC, electron transport chain, in mitochondria. This entire thing is happening is the mitochondria. So this is solely for the eukaryotic cell. In prokaryotes mitochondria absent. In eukaryotic cell, in mitochondria is performed. I am repeating what you need to remember. Pyruvic acid will form acetyl-CoA. It will form citric acid. Then ultimately it's entered the Krebs cycle. Remember the name. You can forget this many names. Ultimate thing is here from we are getting adenosine triphosphate with the help of ETC in mitochondria. In this way, with respiration, we are getting huge amount of the ATP. Okay. Now let's move to our slides.